Next, we'll be learning about ABO blood group. ABO blood group, there are 4 types of blood group. Blood group A, blood group B, blood group AB, and blood group O. The red blood cell of blood group A tend to have what we call antigen A. Antigen, normally we capitalize it. Say this is antigen A, triangle. Red blood cell B has antigen B, semicircle. This is triangle. Blood group AB has both antigen A and also antigen B. For blood group O, there is no antigen, 0 antigen on the red blood cell. I mean, they have other antigens, but they don't have antigen A or antigen B. While this one have both semicircle and also triangle. Semicircle, triangle. So this is what happened. Your blood plasma contain antibodies. Remember I was talking about lymphocytes and how they produce antibodies? Hello?
Yes.
Yeah. So basically, when you're still a baby, you have both the lymphocyte A and lymphocytes B. So lymphocyte A will produce antibody A, and also, lymphocyte A will produce antibody A, lymphocyte B will produce antibody B. So basically, when we were born, all of us have A and B, A and B, A and B. So, So as you age, around the age of 6 months to a few years, that is when your body actively destroy any lymphocytes that will attack or that will kill your own body. So a person with blood group A, antibody A would have caused red blood cell with antigen A to clot. So in that case, your body will remove lymphocyte A. Then there will be no longer production of antibody A. So is B. They will remove cells that produce antibody B. For blood group AB, they will remove both because both of them will be able to tag. Just in case you don't quite understand what is happening, say we have antigen A. Antigen A is triangle in shape, right? Antibody A would look something like this, with a shape that is able to bind to this. Okay? Antibody B, on the other hand, antibody normally have a Y shape it will have semicircle. This semicircle would be able to bind to this. So what's the problem of binding? The problem arises when you have, say you have two red blood cells. This two red blood cell has antigen A. Of course, this person is very likely blood group A. If your body were to produce antibody A, this is bad because antibody A would have caused the two of them to link to each other. Not just one, but probably more red blood cell. Another one over here. So this will form a large solid that is insoluble. Okay, you can imagine this like a blood clot. So there will be blood clot in your blood plasma. This blood clot in your blood plasma will not be able to pass through the narrow capillaries, and it will cause massive jam in the capillaries. Then your blood cannot pass through into your capillaries. There will be a lack of nutrients and lack of gases. You understand what I mean? Lorraine and Tao? Any question? You can ask question.
Then what is both antibodies?
Sorry?
Has both.
Your voice is breaking.
Do all have both antibodies?
Like O has two antibody. It has antibody A and antibody B, but it doesn't have antigen in here. It doesn't have antigen A, it doesn't have antigen B. So it's fine for it to possess both A and also B, because it will not tag its red blood cell. The red blood cell don't have any of this.
Yeah, but they say O is like universal donor.
Yes, it's a universal donor.
But the antibodies can
When you donate blood, you donate your red blood cell. Basically, the blood that you draw out, the person in the blood bank, they will separate red blood cell and antibodies away. So when you were to donate blood to someone else, very likely you are donating red blood cell only. The blood plasma that contain antibodies A and antibodies B probably has been removed. Hello? Even if there is no removal of antibody A and antibody B, there's only small amount of antibody in a large pool of blood. So in that case, it will not cause any havoc.
What's the point of
recipient. What's the point of? Tao?
what's the point of antibodies?
What's the point of antibodies? Antibodies will tag foreign particles. In this case, it just happened that it tagged our own antigen. Antibodies normally is used to recognize, you see the shape over here. Sometimes it has funny shape like this. So if a bacteria is of this shape, then it will be recognized by it. Then it will tag it and show it to the phagocytes. Then the phagocytes will remove them.
Is there like any like facts that because AB doesn't have any
Tao, your voice is breaking. Can you type instead? So basically, just now you were asking, what's the function of antibodies? My answer is the antibodies will tag the foreign particles. Okay, so when it tag foreign particles, it will remove the foreign particles. Then your next question would be, since it tags foreign particles, how come it is tagging your own body cells? So remember, your own body cells tend to have antigens. If you have antigens, they tend to have proteins that are shown out of the cell. So it's some sort of like a form of cell recognition. So this form of cell recognition will make us unique from each other. That's why when you receive organ from a foreign, from someone, even though the organ is useful, but it will probably get rejected after some time because your body's immune system will produce antibodies that will tag against the tissue of the foreign person, because the tissue or the organs is not originated from yourself. So in that case, it will cause your body to reject, because your antibodies produced will be able to recognize the uh, organs or tissue as foreign. Any impact for AB? Okay, so blood group AB, right? It only doesn't have antibody A and B. Doesn't make it a weaker blood group, because antibody A and antibody B is there to recognize your own red blood cell. You have all other antibodies, antibody C, D, E, F, all the way to Z, and then alpha, beta, all this. Okay? There's no impact for blood group AB without antibodies A and antibody B. Because after all, A and B doesn't recognize virus or bacteria. It just happens that it recognizes the protein found on the red blood cell. That's all. Yeah. Okay, so we will proceed to the recipient and also the donor. When you're looking at donor, you have to focus on the red blood cell. Recipient, you look at the blood plasma's antibody. So let's start off with recipient. Recipient of blood group A. Alright, this is A, this is B, this is A, B, and this is O. A recipient of blood group A has antibody B. So if you have antibody B, you cannot have anything with antigen B. So in that case, both B and AB cannot accept. Since you have antibody B, you cannot accept antigen A and antigen, you cannot accept antigen B and antigen B. So in that case, you can accept A and O. If you are a recipient of blood group B, recipient of blood group B has antibody A. So antibody A cannot match with antigen A and antigen A. So in that case, A and AB out. So antibody A can work with this, can also work with this, because it doesn't have any antigen. So it's B and O. Recipient of blood group AB does not have any antibody. So in that case, it can accept everything. Whether you have antigen A, antigen B, antigen AB, and no antigen, they can accept all. So AB is a universal acceptor, universal recipient. Blood group O has both antibody. So in that case, antigen A cannot, antigen B cannot, antigen AB cannot. So only accept from O itself. The rest cannot. So in other words, AB is a universal recipient. O is a universal donor.